Prossimo speaker,
The next speaker, Professor Vincenzo Nigro, full professor of medical genetics at the University of Naples and researcher at the Territorial Institute of Genetics and Medicine, the TGEM in Naples. Dr. Nigro also has a history on sarcoglycanopathies and also on the development of some animal models that allow to study this disease. He contributed to several discoveries, particularly 20 different genetic diseases to define their cause, and he is the coordinator of the Mutation Detection Core Facility of TGEM in Naples. Well, I will speak in Italian, actually, because I was asked to do so. I hope I will speak slowly, so I hope the colleagues with the translation can actually follow me. Well, the main point to have an effective gene therapy is considering the enormous muscular mass that may reach to almost 50% of the total body weight in an adult, between 30-35% in a normal person. And many muscular diseases have the problem of asymmetries in the strength of the muscles that make it difficult to hold a correct posture. So we actually immediately approached a systemic approach, actually. Why? Because the ideal therapy we should implement for muscular dystrophies should be that of recovering strength in a balanced way in the muscles and in the forms involving cardiac involvement to prevent the cardiac lesion, improve the respiratory function, and the therapy should also avoid adverse immunitary response and be long-lasting. You have already seen this chart. Sarcoglycans are a peculiar group of muscular dystrophies. They are not among girdle forms one of the most frequent, but among the severest forms they represent a very important slice of their number. And this slide, I remember there was a job when we actually found a delta sarcoglycan. We defined this complex that has a functional unit. So when we talk about each single sarcoglycan, we also talk about all the other ones. Because if one protein lacks on a sarcoglycan, we do not even find the others that are part of the complex. There are two more sarcoglycan, epsilon and Z sarcoglycan, that replace some of these sarcoglycans in other tissues. By identifying the delta sarcoglycan, we found this form of girdle dystrophy, which is more rare than the others. It is defined as 2F, where there is also a form of cardiomyopathy associated to the mutation of the delta sarcoglycan. So the heart, in the end, is an important target as regards this pathology. And we also saw that in other sarcoglycanopathies, with the exception of the alpha one, there is an involvement of the heart. Patients worldwide that were diagnosed are about 500. Who were made the molecular diagnosis of. I don't know whether this figure has actually increased with increased diagnostics in China and with the full nitrophone sequencing. However, many of these people have series of problems, for instance, the destruction of muscle fibers in time. And when a sarcoglycan goes lost, there's secondary loss of the other sarcoglycans. The animal model we have studied at the very beginning is this hamster, the golden hamster, Syrian hamster, which is a little bit bigger than the mouse and gives us a different point of view compared to gene therapy in mice. A different model, and we can compare the same results we have with the mice, comparing them in a different species. Moreover, of this model, there is a hamster that is called Bio 14.6 that was generated back in 1962. We actually didn't know what the genetic mutation originating a muscular dystrophy was, but more than the muscular dystrophy, the hamster was important for the cardiomyopathy associated with it. So cardiomyopathy connected to the muscular dystrophy. Consider that the quotations, just through a scholar research, there are over 1,000 items on this animal model and over 27,000 citations. It is also one of the most studied models for pharmacological purposes, so not connected to gene therapy. This hamster dies when it is about 11 months old, so with a very reduced life expectancy due to the cardiomyopathy, which is very considerable. What's the advantage of studying this hamster? When we actually implement gene therapy, you can see both the cardiac situation also throughout treatment through echocardiograms that are regularly made, and then you can see life expectancy. So you have an immediate feedback of the gene therapy on this animal. We identified a cause, we found it was a delta sarcoglycan, and we actually applied therapy based on systemic injection of AV, thus putting the human delta sarcoglycan into the hamster. As a matter of fact, this is possible because the human delta sarcoglycan and the hamster one are very similar, and we found a vector that was immediately to be used. We actually used several capsid, which is an external structure that may actually, so to say, package the single helix genome of the virus DNA. And this capsid, we found a series of them for the systemic injection into the hamster, and we tried with both single injections and intravenous injections and intramuscular injections. And we also tried with some combinations of viruses, in which first we had a treatment with one virus, and thereafter we moved to a different capsid, so that we could have a general overview of the different treatments, which could be the best effective one. This is the expression. I would have lots of slides to show you, but I eliminated some of them because I don't have that much time. The stronger result we could obtain was with capsid of 2-8 time, particularly as regards the heart, while 2-9. And if you use these vectors, you know that 2-9 in mice is expressed at a very high level in the heart. It's a viral vector that is perfect for the heart. In the hamster, this does not reproduce, and the heart actually fails compared to the use of 2-8. So 2-8 appeared to be the best vector, but in time we had a decrease in the level of the virus. So by re-injecting, changing the capsid from 2-8, having the second injection with 2-1, we had, after 6 months and a half from the first treatment, actually a sarcoglycan expression that was quite strong. So, we said that when a sarcoglycan goes lost, all the other sarcoglycans are lost. The point is that when you put in a new sarcoglycan, as the colleague showed, all the others have to be re-put in their own positions. All of them have to reappear. So this slide shows how treatment actually produces an important recovery. We also have the pointer here, okay, of the other sarcoglycans in addition to the delta sarcoglycan within the muscle. And again, the most important thing, the cardiomyopathy, as you can see, the cardiac dilatation and the oxidative calcifications that are present in an affected animal. This is a normal animal instead, and through the different protocols, 2-8 actually improves the situation by the 2-9 is less effective in mice. 2-8 plus 2-1 actually produced a recovery in the heart function in time. Then these are all the echocardiographic parameters, and also survival actually improved, so in general. Then we had another possible experiment. We tried to combine gene therapy with drug treatment, and this type of study show that the hamster has something different compared to what happens in the humans, because corticosteroids that are beneficial to humans do not appear to be beneficial in hamsters. But if you have gene therapy is less effective in hamster. We connected this actually to an effect by glucocorticoids on the levels of immunity response, or as regards the mineral corticoid collateral function and cardiac stress. But all these explanations did not find any positive feedback in other trials. However, as regards this cardiac dilatation, the hamster does not behave as humans. Then we tried to see how the vector was moving. We must be safe and sound that the vector does not move onto other organs, particularly in gene therapy, because we can produce damage together with a benefit. We have already seen that the dosage required for effectiveness is very high. It's 10 to 13th, otherwise we do not have important clinical result. We have gene expression, but not with lots of improvements. So based on some works that had already appeared, like this one, we tried to change the capsid, and by changing the capsid in a certain site, that prevented the capturing by the liver of the vector. And to do that, we use the intraperitoneal therapy, administering AAV in the abdomen. Why in the abdomen and not through the vascular way? Because in the abdomen, you can interact earlier, and you would also have this treatment in uterus. So injecting the virus intraperitoneally might allow us to go on working on other facts. And we actually stress the fact that the liver is an organ that would have captured most of the vector. We did some experiments using a modified vector that is not targeted and that does not reach the liver preferentially, that is AAV28 vector that was best than those used in the first experiment. And we saw in the liver that after 2 months using a high dosage or also medium high dosage, we have an expression of COG and AV in some good quantity, and also the expression of delta sarcoglycan that can be seen at the very beginning. It is not the same level we obtain in the muscle, but AVs are present. While with a modified vector, you see the same dosage from 7.39 to 0.11 copies. So here from 4.27 to 0.04. So by simply changing the vector in this way, we could fully prevent the capturing of the virus by the liver. In the muscle, in the same way, we had a better capturing of the vector and also a better expression of the delta sarcoglycan. So by introducing small changes in the capsid, we can actually better target the virus in the concerned tissue. It is not only a point of the promoter where we want to have our transgene be expressed. The point is that is where the vector goes in the systemic injection. If it concentrates in the muscle, it's much better. In the heart, instead, this type of a vector behaves more or less like the other. So this vector might be used in all those forms of girdle dystrophies where there is no important cardiac involvement, since this vector actually mostly hits targets the muscle, and then the complex expression of the skeleton muscle and the improvement, if we consider this vector again, as regards skeletal muscle pathology, so the muscle is definitely recovered. These are the names of the people who are involved in this project. I think I was actually in the times I was assigned. Thank you.
Thank you very much to Professor Nigro. Of course, we can discuss about this presentation.
Faccio io una domanda.
I would like to ask something, and this question refers also to the other speakers. I don't think that there are any studies about the distribution of these vectors as far as the brain is concerned, the brain tissue is concerned. Is there any type of study about this system? No, they are not specifically related to the brain tissue, and this is of course a problem when we try to treat pathologies in which we absolutely need to reach the brain. So we absolutely need to use different vectors, and so that they can better arrive and better reach the brain. Do you have any other question? Please. Sorry, we need the microphone to translate the question. There are differences between the AAV vectors that properly works in mice and in hamsters. Do you have any ideas about how the AAV vectors work in humans? This is, of course, one of the most important questions. Hamster and mice are extremely different as far as the genetics is concerned, and so if we compare many different species, we have a better idea of how the situation works. So if the 2A gene works properly both in mice and in hamsters, it could also be important to treat humans. When the vector works properly just in one species and not in the other, so we have very important doubts about the efficacy in humans. And even the change that you made as far as the capside is concerned, yes, of course, this type of domain is extremely conservative as far as the capturing is concerned. Please. Some of our patients addressed to your center to have a diagnosis about the sarcoglycanopathy and the gene, but of course there is a long waiting list. What can we do to solve this problem? Of course, we tried to use a next generation sequencing analysis in order to analyze all sarcoglycanopathies, but as far as sarcoglycanopathies are concerned, the most important thing to diagnose the problem is taken a diagnosis from the RNA. In fact, if we analyze these aspects, we can also analyze the alterations that are not very well expressed in the DNA. So the RNA is important because we have all the aspects that we can analyze to diagnose the sarcoglycanopathy, and we made the same thing with other pathologies, and so I think that this is a good solution to go on rapidly. Are there any other questions? If there are no questions, I would like to thank Professor Nigro, and I would like to leave the floor